Welcome back for another deep dive. You sent in a chapter from a research methods textbook all about data analysis.
Specifically, null hypothesis significance testing, or NHST.
Right, and NOV.
Exactly. We'll be trying to unpack some of that today.
Perfect.
Yeah.
So listeners might hear about testing the null hypothesis, but what does that actually mean in practice?
Well, you're basically looking at a pattern in your data and trying to figure out, is this just random chance?
Okay.
Or is there an actual real effect happening here?
So you're assuming there's no difference.
Right. We call that the null hypothesis.
So we're trying to disprove it.
Yeah. It's kind of like playing devil's advocate with your data.
Makes sense. So then we get that p-value. What does that p-value actually tell us?
Basically, it tells us how likely it is to get the results we got,
Okay.
assuming that the null hypothesis is actually true.
Ah.
So if we get a small p-value,
Like less than 0.05 usually.
0.05, yeah.
Then that suggests it's not just chance?
Yeah.
Okay. So there might be something real there.
Yeah, something's going on.
But even with a small p-value, we can still be wrong.
Right. Oh, absolutely.
It's not a guarantee.
Not at all.
That's where those type I and type 2 errors come in, right?
Yeah, exactly.
So remind us about those.
So type I error is like a false positive.
Okay.
That's when we reject the null hypothesis
Okay.
and say there's a real effect,
Right.
but actually there isn't one.
So we're kind of like convicting an innocent person.
Yes, based on, maybe not enough evidence.
Okay, that makes sense. What about type 2?
Type 2 is the opposite.
So that's where we miss a real effect.
Yeah.
Like letting the real criminal go free.
Right. Because we didn't have enough evidence to really prove it.
And those errors have different consequences depending on what we're researching.
Oh, definitely.
Like, for example, a new drug.
Yeah. Imagine if there's a new drug, a new treatment,
Okay.
and we make a type 2 error.
We might be withholding something that could actually help people. Exactly.
But a type I error.
That could lead us to approving a drug that isn't effective
Right.
or even harmful.
That's a good point. So it's really important to think about the impact of those decisions based on statistics.
Yeah, always.
And this also brings up the idea of statistical power, right?
Yeah, definitely.
Which is basically how likely are we to find a real effect if it's actually there?
Exactly. And how do we avoid that type 2 error?
Right, okay. The chapter also mentions sample size.
Yes. Sample size is super important for power.
The larger the sample, the more likely we are to find those effects.
Yeah. Think of it like a magnifying glass.
Okay.
A bigger sample size gives you a stronger magnifying glass.
So you can see those smaller details.
Exactly.
But even when we find a significant result, like a difference between groups, we have to look beyond just that, right?
Absolutely. Statistical significance doesn't necessarily tell us the whole story.
Right.
It's not the same thing as practical significance.
So we need to look at the effect size. Yes, the effect size is really important.
So how big that difference actually is.
Exactly. A small effect size could mean there's a difference.
Okay.
But it might not be very meaningful in the real world.
So it's a real difference, but maybe not a difference that really matters.
That's right.
And that's where those standardized effect size measures come in.
Uh-huh.
Like Cohen's D.
Yeah. For comparing two means.
Right. And Cohen's F.
When you have more than two groups.
Remind me about those again.
Sure. So Cohen's D gives you a standardized measure of the difference between those two means. It's expressed in standard deviation units.
Okay.
So a Cohen's D of, let's say, 0.5 means the means are half a standard deviation apart.
I see. Okay.
And Cohen actually gave us some benchmarks for interpreting those values.
So like small, medium, large.
Yeah. So like 0.2 is considered a small effect.
Okay.
0.5 a medium effect.
Okay.
And 0.8 a large effect.
So even if we find a significant difference, if that D is small, it might not be a very big deal.
Exactly. It might be statistically significant, but not practically significant.
Interesting. So what about Cohen's F?
So that's used when you have more than two groups.
Okay.
Like in an ANOVA, it's a standardized measure of the overall effect size,
Okay.
taking into account all those different groups and the variation between them.
So kind of like Cohen's D, but for more complex situations.
Yeah, exactly. It helps you understand how much of the variation in your data is due to the thing you're studying.
So it's like, how strong is that variable really?
Yeah. I like to think of it as how much oomph does it have?
Okay.
And we can use those same benchmarks for interpreting it. Small, medium, or large effect.
Perfect. So we've talked about comparing two means, but what about when we have more than two groups?
That's where ANOVA comes in.
ANOVA, right, which is...
Analysis of variance.
Okay, yeah. So instead of just group A versus group B, we might have A, B, C.
Exactly.
And we want to see if there are differences.
And ANOVA helps you with that.
Okay.
It looks at all the variation in your data.
Okay.
Some of that variation is just random noise.
Right.
But if our independent variable is doing something,
Okay.
we should see a pattern.
So it's like a signal above the noise.
Yeah, exactly.
Cool. So how does ANOVA do that?
So it gives you something called an F statistic,
Okay.
which is basically a ratio,
Okay.
comparing the variation between the groups to the variation within each group.
Okay.
So the larger that F statistic,
Okay.
the more likely it is that the variation is due to your independent variable.
And not just chance.
Exactly.
Yeah.
And just like with the p-value,
Okay.
we look at whether that F statistic is big enough to be statistically significant.
And if it is, we know there's a difference somewhere.
Yep.
But how do we know which groups are different from each other?
That's a great question. And that brings us to post hoc tests and comparisons.
Ah, okay. But we'll save that for later.
Sounds good.
Okay, so these different ANOVA designs, where should we start?
Well, the simplest one is a one-way ANOVA.
Okay.
That's where you have one independent variable with multiple groups.
Right, like we were just talking about.
Exactly. But things get more interesting when we start adding more independent variables.
Okay, more variables, more fun. What happens then?
Then we're talking about factorial ANOVA.
Factorial ANOVA.
Yeah. So in a factorial design, you've got two or more independent variables,
Okay.
each with its own levels or groups.
Okay.
And this lets us look at not just the main effects of each variable,
Okay.
but also how they interact with each other.
Ah, so it's not just like variable A does this, variable B does that.
Right.
It's how do they work together?
Exactly. They can influence each other.
And that could amplify or diminish the effects, right?
Exactly. We call that an interaction effect.
Interaction effect.
And it can reveal stuff we might totally miss if we just looked at the main effects separately.
So give me an example. How would this work in a real study?
Okay, imagine we're studying exercise and diet and how they affect weight loss.
Okay, I'm with you.
So for exercise, we could have two groups.
Okay.
One group exercises regularly, the other group is sedentary.
Okay, makes sense.
And then for diet, we have two groups as well.
Gotcha.
Healthy balanced diet versus no change in diet.
So we've got exercise and diet, two independent variables,
Yeah.
each with two levels.
Yes.
So 4 groups total.
Exactly. You got it.
And we're looking for those interactions, right?
Yeah. Like, what if exercise alone leads to some weight loss?
Okay.
And a healthy diet also leads to some weight loss on its own.
Makes sense.
But the combination, the interaction, leads to even more weight loss than you'd expect just by adding them up.
Oh, interesting. So 1 plus 1 equals 3.
Exactly. Or maybe even 4.
Wow. That's amazing. So that's what makes factorial designs so cool, right?
Absolutely.
You can find these hidden relationships
You got it.
that you might miss otherwise.
Exactly. Those are the synergistic effects. Synergistic? That's a great word.
And we use specific techniques to analyze these interactions.
Like to really understand those patterns.
Yep.
The chapter mentions simple main effects.
Uh-huh.
What are those?
So those help us break down the interaction effect.
Okay.
So we can see how the effect of one variable changes depending on the level of the other variable.
Okay, so in our example, we could look at the effect of exercise.
Yeah.
But separately for the healthy diet group and the no change diet group.
Exactly. So we're asking, does exercise have a different effect depending on whether they're eating healthy or not?
Interesting. It's like zooming in on specific parts of the puzzle.
Yeah, exactly.
See how they fit together.
And by comparing these simple main effects, we can really start to untangle the interaction.
And understand what's going on.
Right.
This sounds complex, but also super powerful.
It is. It opens up a whole new level of understanding.
And we can answer much more sophisticated questions than just comparing two groups, right? Absolutely. You can get much more nuanced information from your data.
And those confidence intervals are still important here.
Oh, yeah, definitely. Always.
Even in these more complicated designs.
Always. They tell us the range of plausible values for our population means.
So even when we're looking at interactions,
Right.
we still need to consider that uncertainty.
Exactly. It's a reminder that we're working with a sample, not the entire population.
Right. Okay. So factorial ANOVA can help us answer some really cool research questions.
Absolutely.
But before we move on, I want to touch on power again.
Okay, yeah.
It's not just about sample size, is it?
No, it's not just about sample size.
There are other factors that can affect it.
Right. The effect size we're trying to detect is really important.
So if we're looking for a tiny effect,
Yeah,
we need a bigger sample size to find it.
exactly.
Like, it's harder to hear a whisper
Yes.
in a noisy room.
Perfect analogy.
And the alpha level matters too, right?
Yeah, the alpha level, that's our threshold for significance.
All right, 0.05.
So if we set a stricter alpha, like 0.01,
Okay.
we need even more power to reach that level.
So it's like setting a higher bar for what we consider convincing evidence.
Exactly. It's all about balancing those different factors.
Okay, that makes sense.
Okay.
So this really highlights the importance of planning our research carefully.
Oh, absolutely. Thinking about effect size, alpha level,
Right.
and sample size. All of that goes into a good power analysis.
So we've covered a lot here, from NHST to factorial, ANOVA, and power. Are we ready for the final part of our deep dive?
Let's do it. I'm ready to explore some of those more advanced techniques.
Okay, so let's move into some of those more advanced techniques now.
All right.
The chapter talks about repeated measures, ANOVA,
Uh-huh.
and mixed designs.
Yeah.
What makes those different?
So these designs are used when we're looking at changes within the same individuals
Okay.
over time or across different conditions.
No. So instead of comparing different groups,
Right.
we're looking at how one person changes.
Exactly. So for example, let's say we're studying a mindfulness training program
Okay.
and we want to see how it affects stress levels.
Okay.
Instead of having a control group and a training group,
Right.
we could measure everyone's stress levels before the training,
Okay.
after the training, and then again, like a few weeks later.
Oh, interesting. So we can see how their stress levels change over time
Exactly.
for each individual.
And that would give us more detailed data than just comparing groups.
It would. It's more sensitive
Okay.
because we're removing some of that variation between individuals.
Oh, I see. So it's like we're removing some of the background noise.
Exactly. You get it.
That makes sense. So we can focus more on the signal.
And that makes repeated measures designs more powerful.
More powerful.
Yeah, so we have a better chance of finding those subtle effects.
Especially if we're looking at something that might have small effects, but they're still important.
Exactly, right.
What about mixed designs?
So mixed designs combine elements of both.
Independent groups and repeated measures.
So, like, one variable is manipulated between groups, and another variable is manipulated within subjects.
Exactly. So, for example, let's say we're studying depression symptoms,
Okay.
and we're interested in the effects of medication and therapy.
Okay. Yeah.
We could have one group get medication
Okay.
and another group get a placebo.
So that's our between groups factor.
Right. And then we can measure everyone's depression symptoms before, during, and after treatment.
So that's the within subjects factor.
Exactly.
So we could see how both medication and therapy affect those symptoms over time.
And how they might interact as well.
Wow. It seems like those designs can really help us understand these complex research questions.
They can.
But analyzing them must be pretty complicated.
It can be, yeah. We still use ANOVA, but the calculations and interpretations get more complex.
I bet. But luckily, we have software to help us with that, right?
We do. But it's still important to understand what the results mean.
Right, so we don't misinterpret anything.
Exactly.
And those confidence intervals, those are still important here too, right?
Oh, absolutely.
Yeah.
Always. They help us understand the precision of our estimates.
Okay.
And they give us a range of plausible values for those population parameters.
So whether we're doing a simple comparison or a complex mixed design,
Right.
confidence intervals are still our friends.
Always. They remind us not to overinterpret our findings.
It's like they're saying, hey, remember, this is just a sample.
Yeah.
Don't get too carried away.
Exactly. They help us stay grounded.
That's great. So this deep dive has really shown me all the different ways we can analyze data.
Definitely.
It's more than just those basic statistical tests.
It is. It's about choosing the right tool for the job.
And thinking critically about our results.
Absolutely. And there's always more to learn.
Always. So before we wrap up, let's go back to those criticisms of null hypothesis significance testing.
Okay.
The chapter mentioned that the null hypothesis of no difference is probably almost always false in the real world.
Right. Right.
So if there are always these tiny differences, does that make significance testing kind of misleading?
That's a great question, and it's something that researchers have been debating for a long time.
Okay.
It's true that finding absolutely no difference is pretty rare,
Okay.
especially in complex situations.
Right.
But that doesn't necessarily mean significance testing is useless.
Okay.
It just means we need to be careful about how we interpret those p-values.
And consider the effect size and practical significance.
Right, exactly.
So it's not just about whether it's statistically significant.
No.
It's about whether it matters in the real world.
Exactly.
That makes sense. And replication is important too, right?
Oh, absolutely. One study doesn't prove anything for sure.
So we need multiple studies
Yes, with different samples and methods.
to really build a strong case.
Exactly.
Yeah.
That's how we gain more confidence in our findings.
Well, I think we've covered a lot in this deep dive.
We have.
From those basic ideas to some pretty advanced stuff.
Definitely.
What are some key takeaways you'd want our listener to remember?
Well, I hope they see that data analysis is a powerful tool.
Yeah.
But it's important to use it thoughtfully.
And responsibly.
Right. It's not just about plugging numbers into a formula.
It's about asking good questions.
Yes. And understanding the data.
And using those insights to learn more about the world.
Exactly. That's what makes research so exciting.
I totally agree. It's been great exploring this with you.
Me too. Thanks for having me.
And to our listener, thanks for joining us on this deep dive. Keep learning, keep exploring, and keep those questions coming.
Until next time.